Hi guys, it's Mrs. Shannon. Today we're going to be looking at the scientific method and we're going to go into detail on experimental design. You will need two things. You will need a pink sheet called the research design table. I have them in the classroom, hard copies. If you don't have this, you can add the notes yourself, but it will be easier to go ahead with the layout I have. The first step today was to go ahead and tape it into page 8 of your notebook. So you will need your notebook, open up to page 8, and you'll see that it says experimental design cheat sheet. What this is going to be is a little cheat sheet of some of the terminology that you'll be using in experiments and come across in some that you read. And it breaks down what each of the different types means and little tips on how to identify them. So really quickly, we're going to go through this slide presentation. And I'm only going to go through the experimental design part in this section. There will be another video on how to apply it to the duckweed lab. Okay, so the scientific method, the first couple of slides talk about how it's a process. It goes in steps. And on page 7 of your notebook, we already covered the steps of the scientific method and loose detail. We also call it the problem-solving method, so you'll just want to review those. Today, we're taking a closer look at the terminology. So this slide right here tells you to fill in the notes, which are going to be in red on these next slides, for each of the next slides in the appropriate box on your research design table. I'm going to go ahead and put this in large screen mode so it's easier. So you'll see that the first box up here in the pink table is hypothesis. So when you come to the slide that says hypothesis, the notes that are in red are what you want to make sure that you include when you're writing on this sheet. So the first section, hypothesis. The hypothesis, we talked about it being an educated guess. The key here is that it's testable about the relationship between independent and dependent variables. Don't stress about these terms. We'll come to it in a couple of slides. The cheat sheet way to remember this is that we want to write the hypothesis in the if-then format. If blank, then blank, and it must be testable. And actually, I'm going to change the slide so that way it says if the IV, then the DV. What this means is that once we identify the independent variable IV and the dependent variable DV, we can just plug them into this format and it will give us our hypothesis. Again, don't stress about the variables. We're going to come to them in the next slides. Let me go ahead and large screen it again. So I know while you're watching this, it might have paused for a second. So the independent variable is the one that's manipulated. It's a factor that it's intentionally varied by the experimenter. The word manipulate means to change. So the IV, the independent variable, is what you, or if you're reading about someone else's experiment, what the experimenter changes. Now the key here is that you can only change one thing, one independent variable. If I go too fast, you can always pause this presentation, go back, see what you missed, and write it in. The dependent variable, so on your pink sheet, we had independent variable, and hopefully you can see this in one box. And now we're going to go on to the dependent variable, which is below it. The dependent variable is the responding variable that is the result of changes made on the independent one. That's all fancy talk for basically saying it's what changes or is measured in the experiment. So this in black is a technical definition, and in red, I sort of gave you some key words to identify what it means. In the dependent variable box, you're going to see two words, quantitative and qualitative. Please write the word numbers with quantitative. You can remember that quantitative data and quantitative variables are ones that use numbers because quantitative has the N in the word. On the other hand, qualitative are non-number measurements, such as the color of something. If something feels warm or hot, but you're not actually using a thermometer, that would be another example of qualitative data. There's a section in the design table. You'll always want to have some background. And for the duckweed one that we're doing, you've researched duckweed a little bit already, and I'll give you some more information. But it's the important parts of the background research that guide your choice in variables or your hypothesis. You don't have to include all of your background here, all of your research, but it's the important parts that are going to come up in your experiment. The constants in the experiment are all the factors that the experimenter attempts to keep the same. You want to keep everything the same for every single test that you do, for all groups being tested, except for the one independent variable you change up. Oops. So examples of that, if we are comparing two loaves of bread, one's made with more sugar and one's made with a normal amount of sugar, the independent variable would be the change in the sugar amount. The constants would be the oven temperature, type of pan, amount of humidity, amount of the other ingredients, etc. The control group, and I know the box in here is very tiny for this amount of red. You can always write it below the box. That's okay. But you want to know that the control group is the group that we're comparing the experiment to, comparison. Usually we can use words such as no treatment, or it's under normal conditions. And I say normal in quotation marks because normal means typical. And what's typical for one organism might be different than for another organism. So when we say the typical environment, where it's found in nature. The control group is exposed to the same conditions as the experimental group, except for the variable being tested. What that means is that the constants stay the same. Now, the experimental groups are any groups that receive treatment, and it could be differing amounts of treatment too. So you might have more than one experimental group. For duckweed, we're just going to use one experimental group. They are the ones manipulated by the experimenter or changed. So if we're doing, we're baking different loaves of bread, and we're using the amount the recipe calls for of sugar, and then we're doubling that amount of sugar. The experimental group would be the bread with the doubled amount of sugar. So I'm going to stop here. If you have any questions about the experimental variables and some of these terminology or terms that we've used, please review back into it. I do have a SpongeBob practice, so you can test yourself. It is available online. You can watch the second screencast for getting ready to do the duckweed lab and how to apply some of these terms and identify independent variable. And it will take just a second for me to turn this off. So yes, I know you're still looking.